Welcome to this lecture on microarray techniques in computational biology. Imagine if you could look inside a human cell and instantly know exactly what every single gene is doing at that exact moment. For decades, scientists could only study one gene at a time, which was like trying to understand a massive symphony by listening to just one instrument. Today, we are going to explore a revolutionary technology that changed everything. We will learn how microarrays allow us to take a massive, simultaneous snapshot of thousands of genes at once, and how we use mathematics to turn those biological snapshots into life-saving insights for diseases like cancer. Let us dive in. To begin our journey, let us imagine you are standing in a massive library with tens of thousands of books. If you wanted to know exactly which books people are reading right at this very second, checking each book one by one would take a lifetime. It is practically impossible to get a quick snapshot of the library's overall activity this way. In the human body, our cells are exactly like that library, and our genes are the books. A microarray is a brilliant technological tool that acts like a massive set of security cameras. It allows us to see exactly which gene books are open and being read by the cell all at once. Instead of testing one gene at a time, we can look at thousands of genes simultaneously to understand exactly what the cell is doing at any given moment. Now that we understand the purpose of a microarray, let us look at what it actually is physically. A DNA microarray, which is often called a gene chip, is a specialized laboratory tool. It consists of a small, solid support, usually a glass slide or a silicon thin film cell. Onto this tiny surface, scientists attach thousands of tiny spots of known DNA sequences. Each of these microscopic spots contains picomoles of a specific DNA sequence. We call these sequences probes, and they represent different known genes. Think of these probes as highly specific molecular magnets, each designed to catch the active signals from one exact gene out of the thousands present in a biological sample. This physical chip is the foundation that makes high-throughput genetic analysis possible. With the chip ready, how do we actually test a cell? First, we extract RNA from a cell. RNA is the messenger molecule that proves a specific gene is currently active. Because RNA is fragile, we convert it into a more stable form called cDNA, and we tag it with special fluorescent dyes. Next, we wash this glowing liquid over the microarray chip. The tagged cDNA will naturally stick or hybridize to its matching DNA probe on the slide, fitting together perfectly like a lock and key. Finally, by scanning the slide with a laser and measuring the brightness of the glowing spots, scientists can precisely quantify how active each specific gene is in that biological sample. The brighter the spot, the more active the gene. Understanding this process is fascinating, but why does it matter in the real world? It matters because of complex medical challenges, like diagnosing and treating cancer. In a healthy cell, certain genes are turned on to keep things running smoothly. But in a cancer cell, the instruction manual goes wrong. The cell starts turning on harmful genes and turning off protective ones. Microarrays allow doctors to compare a healthy tissue sample directly against a diseased tissue sample on the exact same chip. This lets them spot exactly which genes are misbehaving. This incredible technology paved the way for personalized medicine, where treatments are tailored to the specific genetic profile of a patient's unique tumor. Without microarrays, discovering these complex genetic patterns would take decades of tedious experiments. To make sense of these glowing spots, we need mathematics. Usually, the experimental sample, like a tumor, is dyed red with a chemical called Psi5. The control sample, like healthy tissue, is dyed green with Psi3. We calculate the raw expression ratio, denoted by R, by simply dividing the red intensity by the green intensity. In this formula, I Psi5 stands for the fluorescence intensity of the red dye, representing the gene's activity in the diseased cells. I Psi3 stands for the green dye, representing activity in healthy cells. If the red intensity increases while the green stays the same, the ratio R becomes larger than 1, meaning the gene is more active in the disease state. If the gene is less active in the disease state, the ratio becomes a fraction between 0 and 1. This raw ratio formula we just saw leads us to an important mathematical problem. Working with raw ratios can be mathematically awkward and highly misleading when plotting data on a graph. Why? Think about the scale. If a gene is upregulated, meaning it is more active in the tumor, its ratio can scale infinitely from 1 upwards. It could be 2, 10, or 1,000. However, if a gene is downregulated, meaning it is less active in the tumor, its ratio is squished into a tiny mathematical space between 0 and 1. A gene that is 10 times less active has a ratio of 0.1. This creates a severe asymmetry, making it very difficult for computational biologists to visually compare genes that are turned up versus genes that are turned down. To fix this severe asymmetry, computational biologists almost always use a logarithmic transformation. Specifically, they apply the base 2 logarithm of the raw ratio, which is universally known as the log 2 fold change. Here, the symbol LFC stands for the log 2 fold change. By taking the base 2 logarithm of our raw ratio R, we create a perfectly symmetrical scale centered around 0. If a gene is expressed exactly the same in both samples, the ratio is 1, and the LFC becomes 0 because the log of 1 is 0. If a gene is upregulated by a factor of 2, the LFC is positive 1. If it is downregulated by a factor of 2, the LFC is negative 1. This beautiful mathematical transformation makes it incredibly easy to spot significant biological changes at a glance. Before we move on, let us see this with a concrete example. Suppose a researcher is studying a specific gene suspected to cause rapid cell division in breast cancer. The scanner reports a red intensity of 3,200 units for the tumor and a green intensity of 400 units for the healthy tissue. We need to find the log 2 fold change. First, we calculate the raw ratio by dividing 3,200 by 400, which gives us 8. This means the gene is producing 8 times as much RNA in the tumor. Next, we convert this to our standard LFC metric by taking the base 2 logarithm of 8. Since 2 raised to the power of 3 equals 8, our final answer is exactly 3. Because this is positive and large, the gene is highly upregulated, making it a target for new drugs. Now, let us look at another example from the same experiment. The researcher looks at a tumor suppressor gene, which normally protects cells from turning cancerous. The scanner shows a red intensity for the tumor of 250 units and a green intensity for the healthy tissue of 1,000 units. We start by finding the raw ratio, dividing 250 by 1,000, giving us 0.25, or 1/4. The protective gene is operating at only 1/4 capacity in the tumor. We then take the base 2 logarithm of 0.25. Since 2 raised to the power of negative 2 equals 0.25, our LFC is exactly negative 2. The negative sign clearly tells us the gene is downregulated, meaning the cancer has successfully turned down the body's natural defense mechanism. Now that we have calculated the math, let us visualize the actual results on the chip. Here, we see a schematic of a microarray fluorescence grid. On the left, we have our healthy control cells, dyed green, and our experimental tumor cells, dyed red. When mixed and washed over the chip, they compete to bind to the spots. Look at the grid. A red spot means the gene is upregulated in the tumor. There was more red messenger RNA to bind. A green spot means the gene is downregulated in the tumor. The healthy green RNA dominated. A yellow spot appears when both red and green bind equally, meaning the gene expression is unchanged. Finally, a black spot means neither bound, so that gene is completely inactive in both samples. This single image captures the activity of dozens of genes simultaneously. We have covered a lot of ground today, so let us review the most important ideas. First, remember that microarray technology revolutionized biology by allowing us to take a massive, simultaneous snapshot of thousands of genes at once, rather than testing them one by one. Second, by tagging healthy and diseased samples with different fluorescent dyes, we can directly compare their activity on a single chip. Third, we use the log2 fold change mathematical formula to convert raw light intensities into a perfectly symmetrical scale, making it easy to identify upregulated and downregulated genes. Finally, this powerful combination of biology, optics, and mathematics forms the foundation of modern genomics and personalized medicine, turning millions of raw data points into life-saving biological insights. You now understand the core principles of microarray techniques.